stock. Oh. I know how to say that.
I know. I like poking holes. Money. I said money. I don't think so.
No, I had a certain option. Like, we have done it, and then I didn't. I haven't been. No, I don't have.
Okay. So when we left off last time, we got through most of our action potential. So when we left off, we were talking about the refractory period. We're going to go through a couple questions, revisiting these main phases of our action potential, so we can check that we're all more or less on the same page. So this picture is in your slides. I believe it's on slide 17. But if you don't have those with you, the next couple of questions that we're going to do are going to refer to this red line that we're looking at. So that membrane potential change, that is our action potential. Okay, and we're marking a couple of time points. So we got time point A, time point B, time point C, point D, E, and F. So if you don't have that with you or can't find the slide, just go ahead and copy down that curve so that you know what I'm going to do. Copying it. It's a good time to think about what's going on. These different spots. Then a couple of which questions. In here, you're only a little bit, it does not mean graded, it just lets me add more questions in a group. Next, kind of going up. So let's get to the questions. Have you them? and find the depolarization phase of that action potential. Okay, so perfect. Yeah, in that graph, we had A to B showing us the graded potentials leading us up to threshold, and then B to C is the depolarization phase going up to that peak of the action potential, where we see a spike up to positive 30 in our membrane potential due to all that sodium flooding in. All right. So next one, let's find the repolarization phase. Okay, great. So the repolarization phase of that action potential is C to D. As we start to fall back down from that spike to positive 30, we're going back down towards negative 70, repolarizing as that inflow of sodium slows and then totally stops because that inactivation gate is swinging closed. And we have our potassium voltage gated channels opening. So potassium is able to start rushing out of the cell, pulling us back down into that negative zone. Okay. Looking at those time ranges, where do we want to know? Where do we have that neuron hyperpolarized? Yes, so that hyperpolarization, we have just walked through these phases of the action potential in order. The hyperpolarization, when we're down below negative 70, eventually we will come back to our resting membrane potential. That is correct. All right. So speaking about these membrane potentials, when we're looking at that action potential over time, we are just looking at changes in the membrane potential. So we want to be able to identify when we are at rest and when we're not. Okay. So, yes, so we both begin at rest and then end at rest. So we are at -70 at our resting membrane potential to begin with. We have graded potentials forming as we start to have that neuron have signals coming in. We hit threshold, trigger the action potential, spike up to +30 during the depolarization phase, sink down past -70 as we flow through the repolarization phase and into this after hyperpolarization. Then eventually we have that sodium potassium pump equaling everything out, putting us back at our resting membrane potential so that we can do it all over again. Okay. So getting to those final topics we were talking about after going through the phases, left off thinking about refractory periods. When would the neuron be completely unable to fire off a second action potential? A little more of a mix, but the answer is B to D. So this is our absolute refractory period. That's happening first. So during our absolute refractory period, we can't fire off a second action potential first because we're already still working on the first one. And then second, because we have that inactivation gate on those sodium channels closing and nothing can make them open again until they've reset to their original positions. So that's our absolute refractory period during the main part of our action potential. Which leads us into the next question. We have a time phase where our neuron can fire an action potential, but only if a really strong stimulus comes in, stronger than usual. Sorry, it is D to F. I clicked on the wrong one. So D to F is correct. We're talking about sorry, that freaked you out. So this is happening during our relative refractory period. So it's during that hyperpolarization at the end when we are down below our resting membrane potential. Okay, so we could potentially still open some of those sodium channels as they're starting to reset, but it would be hard because we still have those potassium channels open. We still have potassium leaving the cell relatively rapidly. So we have a winner. Except we have a lot of winners. All right. So to recap those refractory periods, right? We're thinking about the action potential. We have an absolute refractory period first, that one shaded in pink or beige or whatever color that is, due to the inactivated sodium channels. And we have a relative refractory period later on, near the end of our action potential, where it would be quite hard to fire off a second action potential, but we could do it as we have some of those voltage gated sodium channels reset. So those were new-ish topics from the end of yesterday, but I do recommend if you were having some trouble with that activity, that chart I gave you with the time points, I gave it to you for a reason, to be helpful. You want to be able to understand what is happening during each of these phases. You want to understand what's happening to the membrane potential, by which I mean, is it going up? Is it reaching threshold? Is it going above 0 to positive 30? Is it going back down? You want to be able to name those phases of the action potential. You're going to want to know the states of the sodium voltage gated channel and the potassium voltage gated channel. And you're going to want to know which directions sodium and potassium are or are not flow. The combination of using that picture and the tables that have been included from your textbook would go ahead and drill those. Make sure you got them down. You will absolutely have some action potential questions on this exam, which I'm missing this. We've spent a fair bit of time here in this packet. Okay. So absolute refractory period first during this main portion, relative refractory period second during this after hyperpolarization part. So we can look just at those channels as well as looking at the point we're in the action potential. So we can separate that out. Our absolute refractory period being because we have all these sodium channels inactivated. And we have our relative refractory period because we have a lot of extra potassium channels. So these voltage gated potassium channels are still open. They're not closed yet. They're working on it. But while we have extra permeability of this neuron to potassium, we're going to have trouble raising that membrane potential up to threshold because we got a lot of positive charges leaving. Our last topic within the action potential, this is just a poll, you're not going to have to put your name in. We're going to start talking about how we have our action potential being propagated. So how we have it moving down our neuron. I want to make sure that we remember the parts of the neuron, because this is something that's going to be happening like in physical space, right? So we want to make sure that we know what space we're talking about. A second or two, through. All right. So the shape of our neuron is just A. So we have dendrites, so these sort of processes that usually receive signals coming in to our main cell body, our soma. So dendrite to soma. As we have that soma pinch off, sort of pinching off point, this zone here, that's our axon hillock. We care about the axon hillock because this is really where we're going to be triggering an action potential or not. We have our graded potentials summing up. This axon hillock has an awful lot of voltage gated channels in it, and it leads us into our axon. So that's that long skinny bit. And then our axon terminals are at the termination, the end of our axon. Those little kind of like foot processes here. Okay. So as we're talking about propagation of our action potential, what we're zooming in on, a little bit the axon hillock, but mostly we're thinking about this region in the center. We're thinking about the axon, because what we're going to see happen is that we're going to have this membrane of the cell get triggered essentially in packets, yeah, like in little sections. So what we're going to see is that, like, first, we're going to see an action potential happening here. So we'll spike up to positive 30, all the sodium's coming in. Then the action potential will move here to so on and so on and so on down our axon. So think about it like a sports game, right? Like when the crowd does the wave, right? Like that's what's happening here when we are propagating the action potential. So we would expect that same little curve to happen here at 10.1. And then a little bit later, it's going to happen here at the next patch of membrane, next patch of membrane. So that's how we have that flow of communication over this long distance. So that's what we have in this final topic. So when we say the conduction of nerve impulses, what we're talking about, conduction, is that movement of the charges down this physical space. So our graphs to this point, including the one that we were looking at in that little practice quiz at the beginning, that was looking at like just a single patch of membrane, looking over time, what would be happening to the charge, what would be happening to that membrane potential. When we're thinking about conduction, we're thinking about what's happening in space. So we're going to see that spike of positive 30 first happen here, then here, then here, then here, as we're going down the axon, from that axon hillock all the way down to the axon terminals. So when this action potential happens at any point in the membrane, so starting out at that axon hillock, and then the first parts of the axon, right, we're zooming in here. Right, so when we have an action potential begin, just in this patch of membrane, what we have happening is that sodium ions are flowing in right to that part of our cell. Right, so this patch of membrane is undergoing depolarization as those voltage-gated sodium channels are opening up, letting sodium flow into the cell. Now, happens to trigger this next patch of membrane, right? So once those sodium channels, once those sodium ions are in our nerve, they start to spread out, right? So this is just like diffusion happens anywhere else. You got them crowded in a small space, they just kind of start to drift, right? Start to drift out to the sides. Okay. So as they start to drift to the neighboring section, so if you have a sodium ion inside this patch, it's drifting over. So we have a sodium ion now kind of near this patch. This has the effect of now depolarizing that next section of membrane to threshold. And so what's happening, instead of having a graded potential come in, which is what's happening in like the dendrites and the soma, or adding them all together at the axon hillock, right? Now that change in membrane potential, that initial change that fires the action potential, is happening because we physically just have some sodium ions floating a little bit out to the sides down the tube. But that membrane is just the same in that patch as it was in the first one. So it doesn't really care why there's more positive charges there. It just cares that we got to negative 55, which is our threshold. So all those voltage gated sodium channels start opening in that next patch. Okay. So we have our next patch of membrane triggering off that action potential. So now our action potential is happening here in this second patch of membrane. This is happening at the same time as our original patch is repolarizing. So we can see in time, right? We have this first patch spike up to positive 30, depolarization. Now it's sinking back down as potassium leaves, repolarizing and then hyperpolarizing, and then eventually coming to rest, right? So we can track what's happening to one patch through time. See what's happening in the neighboring patch as well in this diagram, right? So we have action potential now here in this second overlapping with that same time point where our first patch is coming back down to rest. So we're just going one by one, kind of shooting off down the axis. So the stimulus for the action potential in like one of these central portions of membrane would just be an action potential happening at its neighboring little section. So we have different types of neurons, and neurons can be classified in a number of different ways. Right? So sometimes you'll see neurons classified like by their shape. There are different shapes that they can have. We're not going to worry about that for the purposes of this class. But what we are going to worry about is this myelination. Right? So notice that we're talking here about an unmyelinated neuron. So myelin is like a protective sheath wrapped around a neuron, wrapped around its axon. So it's like kind of like insulation on a wire. So a myelinated neuron would be like an insulated one. An unmyelinated neuron is just like bare. So there's no like covering over the axon in an unmyelinated neuron. Right. So what we're looking at here is how that propagation is happening specifically in our unmyelinated neuron. Right. So starting at that axon hillock, we trigger off an action potential. This would create positive charges, depolarization at that first site. They spread out to the side. That's what these arrows are doing. So then that second site hits threshold, depolarizes, fires off its own action potential. And then it's going to trigger the next site, site C, and so on and so forth. So in an unmyelinated neuron, every single little patch of membrane has to undergo this process. So every little bit of the membrane is firing the action potential. So this actually is slower than what we're going to see happening in our myelinated neurons. So the myelinated neurons, the ones that have insulation, can kind of jump from patch to patch. So they're going to skip parts of the axon. So they're going to be able to go a little bit faster. So we have slow action potential here because it has to go through every little bit individually. So one thing I want us to note, So one thing we want to note about this diagram, we're triggering off patches of membrane. Next patch is depolarizing to threshold because of that spread of sodium ions. But action potentials do go in a direction. They go this way from that body of the cell, from the axon towards the terminals. They don't flow in the reverse, right? They only go one direction. So part of why that happens, why we can have that direction, is because of the refractory period of our neuron. Okay, so when we have this first site depolarizing, right, we have sodium ions, these positive charges coming in. We focused first on the fact that they're flowing towards our next little patch of membrane down the axon, which they are. But sodium ions don't know which end is the axon terminal. They're just spreading out. So they're actually spreading in both directions. So if we compare, for example, on this figure, figure C, right? We can see those positive sodium ions spreading back to site A, as well as spreading forward to our next site, site C. But we don't send an action potential backwards. We don't re-trigger site A because that site is still in the refractory period. Okay, so that site, even though we start to have some positive charges flowing back towards it as the next capsule membrane polarizes, it doesn't send off another signal because it's still working on resetting back to its original state. So that's part of why these refractory phases are actually important. It's important that they exist, because otherwise, as soon as we start to send a signal, we'd get kind of a mess, right? Like it would start kind of bouncing one way and then back and forth and back and forth. So the fact that we have these refractory periods means that we can have this directional flow. We have one trigger, then one, then one, then one, instead of coming back. All right, so if we're going with the wave analogy, right? Like site A has just raised its hands, it's still lowering them. It's not going to raise them and just jump higher because the neighboring section is. That's how the refractory period relates to this directional flow down. In a myelinated neuron, we have a similar process happening, but happens a little faster because we don't have to trigger every single patch of membrane. So what we're looking at here is still an axon, but we now see myelin on it. So myelin, our insulation, is also happening in little segments. So we can see some myelin here wrapping around our neuron. Then we see a gap, an exposed patch of the membrane. Then we see another section of myelin, another gap, another section of myelin, and so on. Okay. So these gaps are called the nodes of Ranvier. So like every single one of these gaps would be a node of Ranvier. So node, just that intersection point, Ranvier. These happen about every 1 to 2 millimeters, kind of depending on where you are in the body. Okay. So in these myelinated neurons, we have our voltage-gated sodium channels really concentrated at those nodes, because those are the bits that are exposed to the fluid, that interstitial fluid surrounding our nerves, surrounding our neuron. So what we're going to do instead of triggering every little patch is we're just going to trigger the segments that are exposed and have all those extra voltage gated channels. So we're going to just like kind of hop space to space to space. Okay. So this leaping motion is faster than having to trigger every single section in order to get all the way down. And it has a special name, saltatory conduction. Saltatory just comes from the Latin word for jumping. So this is this jumping conduction, this saltatory conduction that happens specifically in our myelinated neurons. So specifically in those neurons that have this insulation around. So here we see a diagram similar to the previous one, showing exactly where the charges are in a myelinated neuron. So you can see here our myelin is purple. Those nodes of Ranvier are that little exposed yellow bit between. Okay. And so we can see that that action potential, that positive charge, is jumping just from node to node. Okay. So we have this directional flow still, but we're not triggering every single patch. Now, the speed with which an action potential flows down an axon is variable. So on different nerves, it's going to happen at different rates. But one of those things that changes our speed of propagation, that speed of the action potential, that speed of the signaling down the nerve, is whether we have myelin or not. So myelinated neurons go faster, unmyelinated neurons go slower. Okay. The other thing that can affect the speed and allow it to vary is the diameter of the neuron, so basically like how fat that axon is, has an effect on how those charges spread, right? So when we see an increased diameter of a neuron, so like we see a fatter axon, that makes it conduct these action potentials a bit faster. So you want to think about like if you're blowing through a coffee straw versus like if you're blowing through a boba tea straw, it goes faster through the boba tea straw. That's what's happening here. So when we have that increased diameter, it reduces something called resistance, which we'll talk a lot about when we get to the cardiovascular system. Take some time and a little bit of physics to explain that, so we don't have to focus on it now. But increased diameter just makes that flow easier, essentially. So our neurons that would be particularly fast are going to be really thick, myelinated neurons. So they're wide, they've got that big wide tube, allowing that flow to happen more quickly. And they have that myelin, that insulation around them, allowing them to do this saltatory conduction from node to node. So an example of a sort of speed you might see there is in a thick myelinated neuron, you go 100 meters in just a second, versus if you have a really skinny, thin neuron, you don't have any myelin on it, lacking that insulation, you can still travel, but in that same second, you probably only go as far as a meter. So different nerves, different neurons occur in different parts of your body for different purposes. So sometimes we're really going to want to have that fast speed. Sometimes we don't need that myelin. We don't want that size, and it's okay if we go a little slower. This is just a breakdown of where you would see some different velocities throughout your body. We're going to begin our so thus far, we've just been talking about a single cell, a single neuron, and what's happening inside that neuron. Like, how are we sending a signal down the physical space that that neuron occupies? How are we sending it from the body, axon to axon? Now we're going to get to the point where we're trying to talk to something on the other end. So now we're going to be zooming in on those axon terminals, those spaghetti fingers at the end of our neuron. We're going to be looking at how they connect with other cells. So first we're going to focus on how they communicate with like another neuron in a chain. And eventually we'll think about how they can also communicate with like muscle cells. And so we're going to spend some time thinking about the synapses. Okay. So big picture, a synapse is just a connection between a neuron, so like between a nerve cell and the cell that's receiving that communication. I'm going to draw an ugly neuron. Okay, and here will be my terminals. Okay, so we're just talking first about, for example, a neuron signaling to another neuron. So our synapse is a physical space where that communication occurs, that connection. So we're talking about like this zone right here. So that connection between the neuron and the cell that is signaled. In our body, our nerves, some of them are in your central nervous system, so in your brain, your spinal cord. Some of them are in what we call the peripheral nervous system, so out in the periphery of your body, so like your limbs, right? Other parts of your body that aren't just in the center there. So in the central nervous system, which is where we're focusing first. So think about like, we're first thinking about what's happening in your brain. Okay? So in the central nervous system, the next cell in the chain is going to be another neuron, so another nerve cell. When we're out in the peripheral nervous system, that's when we're going to think about our nerves talking to other types of structures. So often muscles, but we'll also have nerves going to things like glands. So we'll talk about those special types of connections later on. So when we're talking first about these neurons that signal other neurons, right? So if we have one neuron signaling a second neuron, Okay, so I'll label those just on my diagram as well, right? Neuron 1 and then neuron 2. We have special ways to refer to each of those neurons. So our first neuron is the presynaptic neuron. So presynaptic. So pre, because it's before, the synapse being this physical space, this connection with the next neuron. So presynaptic, before the synapse. And then that second one, we name in that same sort of structure. So it's after the synapse. So it's just the postsynaptic neuron. When we talk about what's happening at our synapse, we're going to be specific and say, like, we're releasing neurotransmitters from a presynaptic neuron. They're being received by receptors on the postsynaptic neuron. So that's why we want to know these terms, so that we can talk about what's happening in each of those cells. So the presynaptic neuron, that first one in the chain, is going to signal to that postsynaptic neuron, and that connection can happen at a number of different points. So most commonly, we think about that signaling as happening at the dendrites or at the cell body, the soma. But we do have other connections. So we will see some presynaptic neurons synapsing with a second neuron on its axon itself. And so those are going to modulate the activity of that second neuron. We'll talk about modulation later on. Okay? Just think about, for now, our drawings are just going to be simply presynaptic neuron coming in and forming a synapse somewhere in this dendrite soma kind of region on our postsynaptic region. If we want to name this type of synapse, we name it by location. Okay, so if we're referring to a neuron that's signaling to a dendrite on the postsynaptic neuron, it's axodendritic. So from the axon terminal of the first one to the dendrite of the second one. If we're signaling to the cell body, we have axosomatic. If we go all the way to the axon of the neuron, so like say we're coming in from the side here or something, that would be axoaxon. So all these words are telling you is that where that physical connection is happening. Most of these synapses are axodendritic, and they're unidirectional, just flowing from the presynaptic cell to the postsynaptic cell. So that's what we're going to focus in. Now, synapses are communication between a presynaptic neuron and a postsynaptic neuron. That communication can happen in different ways. So we can have either electrical or chemical synapses, electrical or chemical signaling. We're usually, tell you up front, going to be talking about chemical synapses, but we do want to know that both exist and kind of understand that distinction. When we're talking about these synapses, we're talking about the function of one neuron or presynaptic neuron being associated with the function signaling of another neuron. Okay? Or if we're out in our peripheral nervous system with a muscle or a gland. So here we see pictures of our electrical synapses up on top versus our chemical synapses on bottom. Chemical synapses is what we're going to focus on more. But zooming in on that electrical synapse, so we can exclude this from our further discussion. In an electrical synapse, we are essentially forming a pore directly through our presynaptic cell into our postsynaptic cell. So this pore that we're forming is a gap junction. So that's a type of cell connection. And so we, I think, mentioned type junctions before, where we have our neighboring cells kind of sewn together. A gap junction allows the cytoplasm of one cell to communicate with the cytoplasm of the next cell. So you can think of it as like kind of a bigger version of a channel protein. It's a lot bigger, but it's the same sort of idea, right? So if we have sodium in the cytoplasm of one cell, it's going to diffuse around And it's gonna treat that cytoplasm of the second cell, just like the cytoplasm of the first cell. So we could have ions move directly from the cytoplasm to a presynaptic neuron to the postsynaptic neuron. Okay. So electrical synapses exist when we have gap junctions, cytoplasm touching cytoplasm through that gap junction, which means that ions in the first neuron can flow directly into the cytoplasm of the second neuron, basically just pushing that electrical signal forward, right? So this is going to basically create just like that propagation of the action potential, right? So a similar thing can sort of happen across the synapse. We just have those ions continue moving. So this creates more rapid communication, because we're going to see that in our chemical synapses, we have to put something into the synapse, into a physical gap between neurons, and then we start it. And it does create slight differences in communication, bless you. So in an electrical synapse, your ions could flow one way, but they could also flow the opposite way. So we can have some bi-directional communication happening here too. Places you tend to see electrical synapses are places where you want really fast signaling, really fast communication. So the retina in your eye, you'll see some electrical synapses. The cortex of your brain stem, specifically parts responsible for breathing, and in the hypothalamus, which is related basically to your hormones. Okay, so some of those places you'll see electrical synapses. But for the rest of this course, what we're really going to be focusing on are our chemical synapses. So our chemical synapses are where we're really focusing in on that connection between a presynaptic neuron, the first neuron in the chain, and then a second neuron, our postsynaptic neuron. Okay. We have multiple connections, which is why you have multiple presynaptic neurons labeled here, but we're first just going to understand one of those connections and then kind of spread out from there. And we have a little gap here called the synaptic cleft in the middle, all right, where we're going to have chemicals. This is why it's a chemical synapse, traveling back and forth. So we're going from a presynaptic neuron, then we have the synaptic cleft in the center, postsynaptic neuron. And this is usually happening between the axon terminal of the presynaptic neuron and the dendrite of our second neuron. Okay. Can happen within cases. We're going to stop here for today, but I'm going to give you something to just take a look at. So to prepare for our next session, I recommend finding this picture in your textbook and starting to tease apart what's going on here. We're going to be diving into this next time. Getting a view of like what these spaces are.